Hello guys, welcome back to Milba Academy, and today we will be talking about the effects of variants on the proteins. So the tool that we're using today is PolyPhen, which literally means polymorphism phenotyping. So in my previous video we discussed using VEP, meaning specifically I wanted to use VEP to see SIFT scores, as the SIFT software has been um, moved over to that tool, to VEP. But for PolyPhen scores I prefer using their own website. So what the tool does is that it basically sees the effect of the mutation on the protein structure and function, meaning the whether the active sites are affected or not, or maybe it is structurally affected. It basically looks at the structure integrity of the protein after the mutation. So in my previous video we discussed this same variant, the QCNQ3 gene, and this is the SNP. And for this video we will prefer looking at this. So this is the change in amino acid, basically at the 601st position of the amino acid chain. This asparagine amino acid transforms into lysine. So we want to see what happens after this change. So for that we will go to PolyPhen, and yeah, so this is the place where you look for the effect on the protein for one protein, right? But if you have a lot of sequences, a lot of proteins, I would prefer that you go to batch query, which looks like this, and it also comes with this template that you can follow to get the results. So basically this is the name of the protein that you can get in UniProt, PDB, etc. This is the position of the amino acid. This is the normal amino acid, the reference amino acid, and this is the change in amino acid. So the I, T, E, and G are basically the code names for amino acids. If you want to see what the code name for your amino acid, you just have to search for it. And these days you can even use ChatGPT, but I prefer using just the search engine. I'm not a big fan of AI, though I do use it sometimes when I really need it. So type in amino acid code, and around here you will get this third image. This shows the change in amino acids, I mean the one letter symbols that we're looking for. But you don't have to do that, because they tell you that N stands for asparagine, D stands for aspartic acid, so we're good on that. So as you can see, it helps us because we don't have to Google whatever. Maybe you don't know what SNP identifiers are. It'll let you know what a SNP identifier is. As you can see, it's giving an example RSID you could add. You can also add protein sequence. So if you don't have the RSID, you can simply add the protein sequence, and that is also pretty easy to get. So for that you'll go to ensemble, write the name of your gene. This was our gene. We'll write the name, and so yeah, as from a previous video I told you guys that I prefer using ensemble because it has everything. It's pretty easy to use. It's not very all over the place because you have everything in one side. You can click on it and it will be here. Okay, So we want to see the protein sequence. We'll click on the protein that is at the very top. Okay, so you might think that nothing has happened, but in fact, if you scroll down, you'll get the protein summary. It's loading. My laptop's really slow. Okay, yeah. So this is the detail of the protein. So we want to get the sequence right. Oh shit, where's the sequence? It's here. So if you click the protein, you get the sequence. Yeah, my bad. Okay, so you get the sequence. We need it in FASTA format. So FASTA format specifically starts with this sign that you're seeing, this symbol. And this is not the FASTA format. It is simply just the protein sequence. To get the FASTA format, what I prefer doing is that I go here. Basically, I click the download sequence, but I don't have to download it. I click this FASTA format because I need it in FASTA format. Sorry, I'm saying FASTA format so much. Anyways, so this is the FASTA format. As you can see, it begins with the symbol that we were looking at in the previous window. So we select it and copy it, and we head over to our query data. We insert the protein sequence, and now we have to write the position. So here, as you can see, this is our position. It is the 601st amino acid change. It changes from aspargine, which is the N, and it changes to lysine, which is K. Alright, this should work. So yeah, I'm trying to get the hang of teaching because I'd love to teach. I think I love to tell people new things. I like to give out information, though I feel like I haven't practiced enough. But yeah, so this is all the old logs that these old jobs that I added. This is my old work. Okay, so right now this is also old. This is pending and running. So this is our job that we sent, and it's still processing, so we'll wait until it's ready. If you want that if you want to see if it's done or not, you simply just refresh it. Okay, it's still pending. While it's pending, I'd like to tell you guys, oh actually I think we got the results. Yeah, so this is our report. As you can see, it gives the description. It tells you that it is a canonical mutation, and this is the function of the gene. It is a subfamily of the potassium voltage gated channel. It has alternative names and all that, and the total length of the amino acid chain is 872. Okay, so this is our result. This mutation is benign. As per our previous results in variant effect predictor in ensemble in my previous video, we saw that this mutation was benign, and our we get further confirmation from PolyPhen-2 that this mutation is indeed benign. Yeah. So what's the difference between HUM-DIV and HUM-VAR? Basically, it is for rare alleles, the one that have been identified by GWAS studies. These are for SNPs. So basically, those alleles that are present in at least 1% of the population, these alleles are very, very rare. And HUM-VAR is for Mendelian diseases, the one that you know are um, abundantly pathogenic. So this is not what we're looking for, right? Because this is not a Mendelian disorder. As we go further down, we have multiple sequence alignment. This basically shows conservation. So what really is conservation? It tells us how different or how similar our protein sequence is with other animals. As you can see, this is feline, and I'm not sure what the rest are, but I think this is mice. Yeah, I know that's mice. So yeah, I'm sorry, I don't really know what the rest are, but as you can see, this is very different from our protein. As you can see, there's many deletions. We're not similar to cats, hence the deletion. And lastly, we have 3D visualization. Okay, it's not here. So for 3D visualization, I think I'd make a separate video for that, because for my 3D visualization I prefer using UCSF Chimera X software. It's pretty easy to download. I'll make another video on that, maybe maybe soon, after we see how to calculate our scores, because we generally calculate scores before we see the structures. So if we are sure about our scores, then we'll move over to our structures. But until then, this is all I got. Okay guys, see you in my next video. Do give me more suggestions what I should teach. Also, I do other stuff, like I don't only teach, I can draw. Maybe I could draw for you guys. I don't know. I can literally do anything at this point. Okay, so yeah, see you later.